This podcast is about the role of citrate as a regulator of both glycolysis as well as de novo lipid synthesis. Citrate, in addition to its role in the TCA cycle as the first molecule made in the TCA cycle, facilitating the oxidation of acetyl-CoA, has at least three important functions. First one is that cytoplasmic citrate is an important regulator of glycolysis. Second one, that citrate in the cytoplasm can also regulate fatty acid synthesis. And the third one, citrate can provide the vehicle to transport acetyl-CoA to the cytoplasm for both fatty acid synthesis and cholesterol biosynthesis. At some point during the well-fed state, the production of ATP and NADH will start to inhibit the TCA cycle at isocitrate dehydrogenase, and because of that inhibition, citrate begins to accumulate in the mitochondria. Citrate can be transported across the inner mitochondrial membrane to the cytoplasm, where it also starts to accumulate and its concentrations will rise. As such, a rise in cytosolic citrate can be considered as an indicator of a high energy status in the cell. The first important effect of citrate in the cytoplasm is on glycolysis. When citrate rises in the cytoplasm, it can allosterically inhibit a key enzyme in glycolysis, namely PFK1, phosphofructokinase 1. Inhibition of PFK1 results in the slowing down of glycolysis, and that can be attributed to high citrate concentrations in the cytoplasm. The second important effect of high cytoplasmic citrate is on fatty acid synthesis. High cytoplasmic citrate can promote the polymerization of the rate-limiting enzyme in fatty acid synthesis, acetyl-CoA carboxylase. Citrate promotes the conversion of the monomer form of the enzyme to the polymer form of the enzyme, and the polymer form is the active form of acetyl-CoA carboxylase. So citrate acts as an allosteric activator of acetyl-CoA carboxylase in the cytoplasm during fatty acid synthesis. A third key role of citrate is that it serves as the vehicle to transport acetyl-CoA to the cytoplasm, where it is needed for both fatty acid synthesis and cholesterol synthesis. Citrate transported from the mitochondria to the cytoplasm can be broken down by the enzyme ATP citrate lyase to produce acetyl-CoA and oxaloacetate. As such, the acetyl-CoA produced in the cytoplasm can now be used for fatty acid synthesis by acetyl-CoA carboxylase, as well as cholesterol biosynthesis in the cytoplasm. The transport of citrate to the cytoplasm therefore provides the source of acetyl-CoA, the important precursor needed for both de novo fatty acid synthesis and de novo cholesterol biosynthesis.